We'll be right back after this important word.
A joint. Ever had one? It really sets you up, especially at breakfast time. That lovely mild flavor. Gets you up quicker than anything, that does. Up and away. A good joint sends you off to a flying start. Puts you way out ahead of the rest of the crowd all day. Gives you energy to keep you going. Joints. Grab a basket and nip along to the grocers.
And welcome to the Operation Grow Show, Season 5, Episode... Ooh. One of them, I forget which one now. Episode 15. I hope everybody's doing well and having a good time. And on that note, let's have a quick whip round, see how everybody's doing. Who's here? Who have we got today? Oh, should we try this immersive scene thing? Let's try this out and see how this works. We'll go with, I don't know how many people here. Let's take a fair. Try that. Okay. So, how's everybody doing? Raptor, how you doing, sir?
I'm doing well. Good to see everyone. Yeah, just been hanging out. Really been losing track of a lot of free time lately. Keeping myself busy with our little project we got going on, Beat, and also too just helping out other people in the community with some questions. Some people are trying to find out more about sourcing. So yeah, I'm getting into it myself with sourcing for my own means. So yeah, it's very eye-opening to understand what's available. So yeah, other than that, just been hanging out. We at the Neighborhood Grow Show just did our second episode, so that was fun. We're going to be moving on to our third on Monday, and it'll be an interview with we'll see if the interviewee will make it in time. They are coming from across the states, so yeah, it'll be very interesting. But I know that everyone will be interested to hear what they have to say. Gonna keep it a secret for now until never really talk to anyone in the group about it when we're gonna announce it. So pretty sure it'll be in a maybe even today, tomorrow. But yeah, I'm super excited for it. But yeah, you can find me at Raptor Grow on Instagram, and Neighborhood Grow Show, 4 p.m. Pacific Standard Time, 7 Eastern, and that's 1 a.m. UK. Sorry, everyone UK. If you are an insomniac, you're welcome. As always, we'll try to pick a better time. But yeah, let's see. That's about it for the updates for me. Got my Scarlet Begonias all harvested. So
Nice.
in 10 days, 2 weeks, everything will be going great, and hopefully I'll be having a monitoring system of some sort coming in for the new flower that I'm sending up. So yeah, lots of upgrades coming in, lots of expansion and growing and trying to learn from there. So hopefully I can bring you along with me. Thank you.
No, excellent. All good. Lefty, good sir. How you doing, buddy?
I'm good, Pete. How are you this week, man?
Yeah, not bad.
Yeah,
we deserve a long weekend ahead.
Yeah, don't have many of those anymore. Busy, man. Busy. Got some folks coming to help me throw up the greenhouse tomorrow. Just trying to finish getting that prepped out. Got some Myco stuff happening this week and getting those projects kind of going again. That's been on pause for quite some time, just with the move and stuff. Had to kind of reset and that kind of thing. But yeah, a lot of interesting things going on. I'm trying to work out a consulting thing. There's some things going on here where
Nice.
I think just try and do some consulting, help people set up equipment, decide what they want to do for a space, help them pick genetics, stuff like that. Maybe just help them kind of get started. But just kind of get my feet wet with that, get an idea going and stuff. But yeah, just, busy, man. Everything is super busy. Got the project going. Stuff coming down for that. I think I'm at 70 days today. I let stuff run a little bit longer. As far as the seeds go, seeds on the tops of the plants look really good, fully mature, nice tiger striping, really good dark, real nice seeds. Halfway down to the lower part, not quite as mature. So yeah, I'm doing seed production, so I'm gonna let them things go until I'm happy with them being mature all the way down the plant. But everything's looking pretty good. Learned a few things this round, and yeah, looking forward to popping some of these F1s and seeing what comes out of them.
Nice. Sounds good. Yeah. I know the Mystery Grow is going to look to be popping some of the F1 seeds from that initial round of pollination coming up soon. So that should be interesting. See if that purpling phenotypic expression they spotted passes down through the line. It's definitely caught their eye. So yeah, things are going to start being interesting. Yourself, Duckweed, how you doing, buddy? If he's still there.
Oh, what up? Cheers, everybody. I'm fine. A rainy day out in the Caribbean here. It's a chilly 28 Celsius.
Oh.
Oh yeah. Just getting my hoodie on and everything. Other than that, not much really. Just harvested some autos.
Nice.
Been coming along really nice. I got like about 80 grams from one. That was dry weight.
Very nice.
Yeah, and yeah, still well, just making more space in the tent to pop some more of those TKM10 Platinum Bypasses. And the what was it? Slurry cane onto GMO map.
Okay.
So yeah, I'll probably be taking a male from the TKM and putting it onto a female from the slurry cane, and male from the slurry cane and a female from the TKM. Both ways.
Nice. Yeah, and you've got hurricane season starting to creep up soon, haven't you?
Not soon. Hurricane season is usually like closer to the end of the year, like September, October.
Okay.
But yeah, we're definitely getting into the rainy season for sure. So I don't know how much I'll be doing outdoors.
Yeah, humidity becomes a problem.
Yeah, for sure. Because like today humidity is like 97% today.
All day.
Yeah. So
that's the low.
So yeah.
Oh dear. GMF, how you doing, buddy? How things going with you?
Oh, I can't complain. Kind of in our humid time here too, so it's just the joys of spring. Not running the air conditioner yet, but the humidity is climbing every day. It's raining every day for the most part, so a little bit here and there. Just the joys of spring. Same thing as everybody else. Got some new stuff popping as well. That PCK by Golden Goat. We'll see how that goes. Making some weird beer beverages as usual. I got a kombucha mom started from just juiced weed leaves rather than using tea. We'll see how those moms start. And trying to make a couple of infused kombuchas and a couple infused low to no alcohol beers. So yeah, my usual stuff. Making bread, making pickles. Nothing exciting.
Nice. All good there. All good. And on that note, Cascadian, welcome again. Well, I'm gonna say welcome again. Do you here on regular on the show? I mean, it's like I'm making a thing of it. But welcome again and doing a presentation for us. And today it's really going to be taking that sort of step back to, in a sense, going back to basics, but more, I guess, in terms of the building blocks, basics, the brick and mortars of the genetics, the actual what's going on down at a much more, not molecular level, but at that cellular level, what's going on.
There we are. We're on live.
Probably a good way of
What's
doing
up,
it.
guys?
Hey. Turn it sideways. Hey, Tanasi
How's it
and
going, y'all?
Keith.
Oh goodness!
Just wanted to hop
Double
in and say
whammy.
hello. Say hi,
Hey.
everybody.
Hey, how you doing?
Pretty good, man. I guess it's the Kentucky Derbies today, so we're gonna get drunk on mint juleps and I guess watch some government sanctioned animal abuse. It's gonna be great. I hope all the horses are on tons of steroids and they run super fast.
Oh shit, Keith, are you
And
gonna
the
be
ones that
passed out on the
don't.
floor shortly?
Oh shit.
You've got your
All right,
glue
anyway.
truck and steak and burger van
Yeah, I
parked
know.
out back.
My phone, guys, my phone is I have an iPhone 6 and it can tolerate this kind of treatment for about 30 seconds before the battery dies. I
No
just wanted to
worries,
hop
buddy.
in and say hi.
No, excellent. It's good to see you guys. Yeah.
Dude, I had so much fun last week. I had to say hi. Tanaz is gonna do a dab for you.
I wanted to say
Oh.
cheers to everybody and dabs up, eh?
Cheers
Hey,
tonight.
dabs up, buddy.
Cheers, Keith.
Dabs up.
Hey, Tanazi,
Dang.
thanks for the fast shipping, buddy.
Say what?
Raptor said thanks for the fast shipping.
No,
I did.
Oh, they're
Lefty
Lefty.
did. that.
shipping out
Lefty.
every
I
day
said
about
cheers.
Monday through Friday. We're in that UPS shipping out.
Real quick, y'all, I got a treat for you.
Right on, right on.
I'm gonna
Oh,
show you guys the mountains of Colorado.
oh. Let me. Oh, we missed it. There we go. Oh no, there we
Can
are.
you see it?
Oh yeah. Oh,
There on the horizon.
oh yeah.
Mm-hmm. Very nice.
Anyway,
I miss being there. Oh well.
all right, y'all.
Soon enough.
Hey, have a great show, man. Thanks for letting me stop by.
No, cool.
Cheers,
Thanks for
guys.
stopping by, dude.
Later,
All right.
Keith.
Peace.
Stay well. Cheers, guys. Oh, and then on that note, back to
Yeah, man.
Cascadia. Well,
Fun fact about the mint. Fun fact about the mint julep. You think the original recipes were actually made using peach brandy?
Hmm.
'Cause that was
I
one
have
of the
the-
first spirits we had here.
I have the julep mint outside. The particular mint that they use to make those, actually. It's
Makes
not
sense.
not peppermint or spearmint. It's an actual different variety of mint. So it's my
Makes
favorite,
sense.
actually.
Yeah, Kentucky being peach brandy and all that
Right.
moonshine running around.
Well, peaches were a
Nice,
native
nice.
fruit even before the apples. So yeah, and they tend to rot really fast. Even when you make them into a hard cider or something, they tend not to last. So distillation it was.
I have a. Go ahead, Lefty. You're saying. Or someone else? Oh, okay. Cascadian, I had a question about the mint julep in comparison to the like a chocolate mint or a spearmint or peppermint. What differentiates it?
It's not. I mean, spearmint or peppermint is like the essence of what it is to be minty. So I wouldn't say it's more minty, but it's more like fresh, like maybe sweet. I'm not sure the right word to apply to. It's got a slightly different profile, but
So it's minty, like intensity up to that peppermint kind of scale, and with like the sweetness of a spearmint, maybe.
it's. Have you ever had lemon balm?
Yes.
It's like the mint. It's like the mint version of lemon balm. It's like it's kind like that. It's really like, it's not overwhelming or overpowering. It doesn't smack your senses around. It's kind of.
I grow a lemon
well-balanced.
balm.
Yeah, it's kind of well balanced. It's kind of it's really just a pleasant herb, man. It's incredible.
Thank you.
Where-
Just hearing
where-
balanced makes
yeah,
me
where
understand better. And
peppermint
it's
is,
not
like,
so
really,
much
like-
peppermint. It's not so much sweet.
Yeah, peppermint's got that like volatile oil like in your nose, in your senses.
Yeah.
It doesn't have that. It's like it's even keeled on that note. So which I think is why I
Balance.
found it so pleasurable, is because it's more. I mean, you can just pop it in your mouth and it's just like, oh, give me some more. Like it's not like
Yeah.
peppermint. We're like,
Drink some water and then like that feels even better. Okay,
Yeah,
good to
it's
know.
awesome.
I'll keep my eye out for it. I'm a fan of the mints.
but yeah, with all the derailing, that was fun. Thanks for Black Sail for popping in. Glad to be here. We're getting back to the intro. Thanks for having me. We're gonna get to the nuts and bolts here pretty quick. So This is yet another presentation you guys will probably have to watch numerous times. Pretty vocab heavy, and gonna try to present this in a way that is approachable. So, you know, stop me if you get lost or there's fundamental questions that I'm missing. You know, let me know here. So today we called this
We'll keep an eye on chat for you.
Back to the Basics Part 1, because there's going to be, you'll see when we get to the end, there might be a few more parts to this. So today we're talking chromosomes, and from there we're going to get smaller. So we're getting into, we've been throwing around a lot of words and they haven't had a whole lot of context the last couple weeks, 6 weeks, a couple months. I'm not really sure at this point. And this is going to apply some context to what we have talked about and what we're going to talk about so that we don't end up with a chat room full of frustration. So chromosomes are something plants, animals, all life forms have. I think fungi have chromosomes. I think so. I think they're just weird. But anyway, pictured on your screen here is a snippet from Molecular Cytogenic Characterization of Dioecious cannabis sativa with XY chromosome sex determination system. This is an excerpt out of the paper. I snipped, these are actual cannabis chromosomes. This I on top is a male. The J on the bottom is the female. And these, each one of these individually is a chromosome, otherwise known as a thread-like structure of nucleic acids. And this is where genetic information is stored. These chromosomes are found in the nucleus. I didn't want to get into a whole cell biology tangent, so just know they're in the nucleus. If you want to know what the nucleus is, I might get there someday, but not today. It's the command center of a cell, basically. It's where all the important stuff happens.
The powerhouse of the cell.
That's the mitochondria, technically.
Oh,
Yeah,
right.
I mean, that's where they make energy, mitochondria. This is where the nucleus is where all of the pertinent information is stored. It is under guard of both the cellular membrane and another lipophilic membrane that makes up the nucleus that is highly selective. So inside of that nucleus is very protected, and that membrane has, it's like the difference between going into a military town and then trying to get into the military base. You know, so a lot of people can get into town. You got to have an access card to get into base. To get into the nucleus, you basically have to have a genetic or a cellular access card. These chromosomes live inside that base. Oh, what I wanted to point out, are we still on chromosomes?
Yeah.
Let's go.
No,
back.
there it is. Yeah, it's moved.
Huh.
Around
It says, what is a locus?
Yeah, okay, here. I wanted to point out really quick, cannabis is what they call, I didn't write it on here, but it's 2N, number 2, letter N, equals 20. So what that means is it's dioecious, 2, and it has 20 chromosomes, 10 pairs. So it gets, if you see here, we've got 1 pair, 2 pair, 3 pair, 4 pair, 10 pairs. The 10th pair is the sexual determination pair. That's what tells you, that's why their XY and the XX are down here. This is sex determination chromosomes. If I remember right from the paper, the red dots are indications of female. I don't want to quote it wrong. I don't know if it was genetics. It's analogous with the female representation, which you'll notice on the bottom, there's only red. And then on the top, we get these greens. and that's a representation of male genetic presence, basically, because I don't want to butcher it. That's an indication. They highlighted them here. That's the indication that there's male genetics present, which tells them that I is a male and J is a female. So I just wanted to lay that out, that there's 10 pairs of chromosomes. Cannabis has 20 chromosomes in it, 10 from the mom, 10 from the dad. So within a chromosome, there are what we call locus, or singularly called a loci, depends on who you talk to. This is a portion of the chromosome where a particular set of information lives that we call a gene. This is how they determine, or how they label where different genes are in the chromosome, is they call them loci. That's how they refer to them. So in the top picture here, you can see that they have a heterozygous and a homozygous locus. So this, if you remember from previous weeks, we were talking about big A, little a. This is what we're talking about. One strand has a big D, the other strand has a little d, and together the expression is heterozygous. When you have two of the same, you get homozygous, but you get one letter, for right now, one letter from each chromatid that makes up the chromosome. I didn't want to lose you in all this, so I'm just starting at chromosomes, and there's 1,000 tangents I could go off with more vocab. I'm just trying to keep us on track here. So individually, they are called chromatids. Together, they are called chromosomes. At each point, they line up. So the second figure here, you can see they line up the locuses, the loci on each chromatid of the chromosome. Vocab days, guys. It's vocab day. They have a different code. They have a different gene. And one of these will play dominant over the other, and you'll end up with, heterozygous. A big D, little d sort of situation with this white flower, purple flower. I'm not sure why they went purple for white and green for purple. Anyway, that's a locus, or that's locus or loci, depending on how you want to say it. Inside of the loci is alleles. When we're talking about alleles, we're talking about one of the two of the loci. So it's just one part of it is an allele. It's not the two together, that's the loci. Individually, they're alleles. The big A itself is an allele. The little a itself is an allele. Together, they are a loci. You're lucky I didn't make like a Quizlet for this and have everybody run through it at the end to see how you guys all graded. But yeah, vocab days, guys. So upper right hand corner, we've got the same chromatids that make up a chromosome, and we're highlighting the loci, the individual loci across both arms, and highlighting the different alleles in this case. These other pictures are further representation of that. If you know, some people are visual learners, some people are audio learners, so I'm just trying to give everybody what they need to pick this up, because I'm just trying to put it down for everybody. You'll see this graph here looks very familiar to some of my previous presentations, so hopefully some of this is able to sink in and you can place it in context of previous conversations. And here is a pretty decent example of how this works across the whole chromosome, even though this is a very basic example with only four loci that they've got labeled with boxes. You know, they've got 6 actually labeled, but there's thousands on each chromosome. So this is just a very, very basic example here to give you guys a representation of how this works. This is very much a 6-hour presentation shoved into about 10 minutes. So for that, I'm sorry, but it's all here. And with enough review of this and enough staring at this and enough questions when I'm done, I think we'll be okay. So we started
And-
with the chromosome. Go ahead.
You're saying then what's on this picture here, in the bottom right-hand corner,
Right.
where you see the gene loci.
Well, yeah.
Anyway, but that's what's interesting. I mean, that's showing that it requires these three loci together to make up the gene. The gene isn't just one of them, so it's these three together in this, and they can move around as well, so that they can shift into a different position.
Right.
Yeah, so it's not one. It's not like what each one of those is a gene. It's a combination of those together form a gene.
Right. So we kind of went down one concept of what is on a chromosome. This is the second half of what a chromosome is all about. So as Pete was referring to, several alleles lined up in a row make a gene. That is standing on the diving board of a really deep conversation that I hope to have another day, as far as how we get from a gene to something we see in the plant or person. That's called transcription and translation. And I think I purposefully left that out of the pictures here. I did have some pictures that showed it, but I don't think I ended up using them. So this is just to illustrate how a gene can be found on a chromosome. So a gene is as it says, the unit of heredity that we track. So you'll have a unique sequence of loci filled with alleles, and there's a lot of examples out there that say this in the context of a sentence. So they use capital letters for, we're going to get to it in a minute, but they use capital letters to signify what is at each nucleotide inside of the allele that makes up the gene. It gets really confusing really fast, but it's like you can change the sentence based on the alleles in the gene. So when you swap the alleles out, what you see from that changes. And that's literally how we get different expressions. Yeah, vocab days. This is just vocab. There's a lot more to get into, because this is, I took a whole quarter of college to really understand all of this. So I feel
So
kind
it's
of bad
like-
slamming it all on you guys in 10 minutes, but
So it's like you changed the meaning of the sentence by just changing where the word, the allele, is placed.
right. So here we're seeing nucleotides. Hope I haven't lost you guys yet. I can't see the chat right now. Nucleotides are, it gets more clear as I get through this. I'm sorry that I'm stumbling, but there's a slide at the end of this that's going to bring it all together. Nucleotides are the blocks within the allele. So now we started at the chromosome. We went to the loci, which is the part of the chromosome. We went to the allele, that is one half of the loci. And now we're looking inside of that allele. And inside of that allele is this sentence, this AUCG that you're seeing on the right side over here. These are the letters making up the sentence. When you swap these out, you change the allele, which changes the expression, which changes what you see. I hope that makes sense.
Mhm.
A nucleotide itself is made up of a phosphate group. Either it's usually a pentose sugar, pentose meaning it has 5 carbons. That's why it's 5-sided here, pentagon, pentose, pent 5. And then a nitrogen containing base. I'm glad I wrote it on the screen because I just blanked. A nitrogen containing base. And these nitrogen containing bases are amino acids. I'm sure you guys have heard that word. There's only 6 amino acids that are used in DNA and RNA to type the sentences. They only have 6 letters. And they don't have all 6 letters in every context. They've usually only got about 4. And it depends on the context as to what 4 they have. So there's only 6 letters in the alphabet, and that makes up everything you've seen in your whole life. That tells everything to code everything that you've ever seen. It's kind of crazy. If it was alive, it played with 6 letters to get there. Why this all matters is because DNA itself is built from, if you guys have seen the double helix part is these phosphate groups. The connectors in between, I think if I hit the next here, yep, all the connections in between are the ribose sugars and the nitrogen bases. So to really get an understanding of how genetics works, you have to wrap your head around this stuff. This yellow band on the outside is the phosphate backbone. On that is always a pentose sugar. But the code that we play with, the sentence that we type changes. And the way that DNA works is that there's 2 of these strands, 2 of these phosphate backbones. And the bridges through the middle create, this is a very simplistic, so the image on the left is more correct in how complex this can get, but even then it's missing some detail, because there's hydrogen bonds, as it says. There's also my chemistry teacher would be mad right now. There's another type of bond. Anyway, there's another type of bond that goes, it's Van something.
Covalent?
Nope. VAN lowercase space. It's somebody's last name, and I'm blanking hard right now. Starts with a W. Van Walls? Anyway, there's another type of bond, and all of these bonds play into the structure of DNA. They play into how the coding fits together, and all of this matters because of that security stuff I was harping on a little while ago. They double check, triple check, quadruple check that this code is right, because when this code changes, you get mutations, you get different expressions. And this code in particular is important because it has a lot to do, van der Waals, thank you, has a lot to do with I had it and I lost it because we got onto van der Waals. What was I saying? Anybody?
On.
It has a lot to do with mutations. If you change the letters, it'll change the expression. You can get a mutation, and you can break. So you can break the proteins. You can make it so that you cannot build. It doesn't work anymore. That gene is broken. It doesn't express. You have a defective protein, that therefore you've lost the ability for that protein to work inside that plant, and whatever that protein did, that ability is gone.
Um, I mean, it's a lot like a instruction book where you can move some of the words around, and you can actually move quite a good portion of the words around in an instruction book before it stops making sense and stops actually working and being able to be used as an instruction set. So, you can swap the occasional word here and there, and that doesn't affect anything too much in terms of what it's at. Another way of looking at what's going on. So how you reorder those words is particularly important. And letters, occasional misspelling.
Right. Which DNA is like double, triple checked for security, but then it's also slightly redundant. I don't want to get into how today because I feel like I've completely overwhelmed everybody. But there's redundancy built in, and there's additional protection measures for keeping this information, to keep the sentence written the way the sentence was wrote originally. There's a lot of mechanisms involved to make sure that it still reads the same, because if it doesn't, life goes haywire and we're not alive anymore because it all breaks. I'm sure if anybody was paying attention and I didn't lose you, there's bound to be questions. I would love to have some questions so that I can synthesize this for everybody, because it really takes a conversation to make this sink in. It can't just be preached to you because it is so complex. But before I get that far, I want to show you how this matters in the future when my slow computer decides to load the next slide. I did click, right? There we go. Oop, I clicked too many times. Future topics. This goes in a bunch of different rabbit holes. Exons and introns has to do with what part of the sentence is actually the sentence, and what part are the periods or the ... before the next sentence starts. That's some of the redundancy, some of the security, is they have blank spots in between the genes that don't read anything. Telomeres, those are blank spots basically at the ends of the chromosomes. And as things are copied and as things are, to be correct, transcribed and translated, those telomeres will get shaved off. And that's thought to be part of the aging process. That's thought to be, because once you start to lose so many telomeres, you actually start cutting off things that matter, and you get to a part where you've broke it so far that it doesn't live anymore. So
They're like a parity check bit. So making sure that replication is taking place correctly. But then every time replication takes place and you do a check, you shave a little bit off. So the next time around, it can't check it quite as well as it did the last time because the bit's missing. For the bit that it had to keep filling in to keep replication on track.
And that comes in a when you're making new cells. I'm a little messed up today, guys, so sorry that my speech is not quite as eloquent as it usually is. I'm having some nerve problems, but telomeres are involved with that process of making sure that as one cell makes more cells, that it ends up with the right, because you have to copy your genetic information to the new cell. You lose a little bit of that in the copy. So a cell can only divide so many times before it's shaved to completion and, for lack of a better term, like bald. You're welcome, Pete. Purines and pyrimidines are the different structures involved with the nitrogen base portion of the nucleotide. So it has to do with whether these have 2 rings or 1 ring and what that means. There's differences in the structure and what those do for DNA. That's what this purines versus pyrimidines is all about. Has to do with the amino acid structure on the nucleotide. Transcription and translation is the act of taking DNA and making more DNA. It uses mRNA and it uses what are those things called? Starts with an R. Ribosomes. Uses a ribosome. A ribosome will come over to DNA, unstitch it. The mRNA will go in there and read it while the ribosome is holding it open, and it'll spit out a new DNA. It'll read the code, and it's like a printer, but it's really like the person in court who's writing down everything as it's being talked. They're just transcribing what's happening and spitting out this new reel of information that then goes in the new cell to make the new cell function. That's transcription and translation. Meiosis is sexual reproduction. It's when chromosomes are brought together and pulled apart, and we can talk about all the mutations and allele swapping and things that can go on in there, which is why you plant 100 seeds. That's why they all look different. It's part of it. There can be some things that go on in there that bring increased variability, unexpected variability. But if you understand how meiosis works, you can account for that. And then recombination. Shit, I just blinked on what recombination even is. It's a great day, guys. And I'm sober, believe that. I told you though, my nerves are shot today. I've been fried for 3 or 4 days now on my nerves.
Yeah, I'm excited for the telomeres. And great presentation, by the way, before we go any further. Very informative. But yeah.
Appreciate it.
Pretty excited for telomere discussion, just because I've heard a lot of mentioning of it, but not so much a breakdown and application of how to adapt it to
Let me kick it over here.
plants and horticulture.
Okay, now I can actually see my slides here, and we can field some questions if you want. I'm sure there should be some. What holds one strand against the other in the double helix? Those are the van der Waals and the hydrogen bonds. It's attraction forces that's holding all of that together. It's kind of hard to wrap your mind around even after you've been taught it in a university setting, because it's just that fascinating that on molecular levels the bonds are strong enough to do that sort of thing. Yeah, so it's attraction forces, and then it's all electrons, positives and negatives, and hydrogen atoms that are causing charges to keep these things held together. And then, I mean, you've got to understand DNA is wrapped up. That's the other thing I didn't put on future talks, is histones and DNA packaging, and this goes on and on and on and on. It's all raveled up in bigger pieces that are then like DNA is wrapped in histones, and then histones wrap in a way that's all convoluted. So there's several layers of attraction forces all holding this together, which is what's so mind-blowing about when a ribosome comes in and says, we're going to open you up and we're going to unzip the DNA and then read it and then make a new portion. Or is that mRNA? I'm getting confused. Maybe it's the mRNA that is.
I believe that is mRNA, messenger ribonucleic
Messenger
acid.
RNA. Right. Yep. You're right. It just came back to me, which is a single strand. DNA is double strand, double helix. mRNA is a single strand. So mRNA comes in, opens it up, and then reads off one of the strands to get the gene that it wants, or the allele, the gene, the expression that it's looking for. It reads that particular part to make the protein associated with that part of the chromosome. And then when it has that protein, it sends it out of the cell. So this is all, I mean, this controls everything in the cell.
One hell of a librarian.
Yeah.
Like,
Yeah,
what do you
yeah.
want again?
Yeah.
Wait 2 seconds.
With a precise pencil that doesn't make an A look like a B or an A look like a C, or, I mean, an A is obviously an A. Like
Yeah.
it's really cool.
Occasionally they slip up.
Which is where mutations come in for sure. Yeah, it's
And then that whole process of sequencing the DNA, or is just understanding the sequence of those letters. That's the sequencing of the DNA. Then mapping the genome is working out, okay, this combination of these letters is what controls this that we see, or produces this protein essentially, which controls this, etc., etc., etc. And that's the bit that takes a long time to pull off.
I hope that made sense, guys, because I really feel like I shot that like fish in a barrel. Like I don't feel like that made any sense at all. So I hope you guys got something from it.
Yeah, no. Work. I thought that was very good indeed.
It's one of those I might have to watch a couple of times.
It's a lot of
But
vocab.
That's a good thing about it though, is there. They keep coming back to always reference, you know. So
yeah, I know somewhere where it's recorded.
And once
It was a good quick refresher for me, you know, from 13 years ago when I took all this stuff at Michigan State. We kind of, you know, did a quick blast through it, and no, I mean you broke it down pretty well. You packed a lot in, but you broke it down really well, I thought.
Yeah, I appreciate it, because I'm trying to give a lecture on things I was taught 4 years ago. So it's even, you know, I had to go back through my old presentations and my old notes to even put this together. Like because there's so much to it. You're gonna lose details. I've over jumped numerous things just to try to get this basis laid out. So I totally feel like I botched it, but I think it's because I know all the other things I left out. That's why I'm looking for feedback.
There's only so
thanks
much
for
you can
taking the
dip
time
into.
and effort to do it. Yeah, man. Like you edified people more than they were before. So thanks for taking the time and effort to do it, dude. That's, you know, and this, like, people don't understand, this is applicable to everyday life, right? We're all talking about a virus, and really, you know, the reason that we're, you know, using this new vaccine technology is it uses mRNA. That isn't how we used to produce them. Once you understand what mRNA is, it takes a lot of fear out of things that are known by just by their acronyms. So thanks for the edification, dude. I appreciate you sharing all that.
Yeah.
Searching for questions, guys. If there's any questions in chat, lay them on me.
Yeah, I think it's just amazing how they get down to the point of, okay, we can come in now and we know this is the bit we're interested in, and go snip, cut that piece out, go and find another bit and snip it out from somewhere else and wedge it back in the way it's supposed to go. And that to me blows my mind when you how they figure that out. It's definitely some seriously smart people out there.
All the questions I see, you've either addressed directly in chat or said it out loud.
Yeah,
Yeah, everyone's saying it made great sense and everything.
Yeah,
So,
I appreciate you guys. Thanks for the the vote of confidence, because from this side it feels just like a mess. But
Well, you know how chat gets sometimes.
Well, the thing is you have all that information in your head, and an in-depth, scale to where you're trying to put it to a layman's terms and jam it all in, like you said, in a half an hour. So yeah, I bet you it is a big mess in your head, but it came out really, really well. So
cool.
I know what you mean. You're trying to explain something from a really in-depth level, kind of like compacted and less scientific and much simpler. So yeah, that can be challenging.
So yeah,
You know how much more detail you can go into.
It'll be really exciting when we get to the point where we can talk about like selecting at one loci or selecting at two loci, because you can do that, or figuring out that two loci, even though they're not on the same chromosome or they're not anywhere near each other, are connected. It doesn't matter where they're at the chromosome. Through breeding and observation and growing your plants, you can start to tell that this is connected to that, because, and you'll understand why. It's because the loci are connected. It's because how the protein folds, and when they try to unfold it and read it, it can't read the one without read the other. It'll be fun to get into sex-linked traits and cross-linked traits, and because that's all you have to understand, this terminology and this vocab to even start to approach that stuff. Recessive traits. Recessive traits come from some of the earlier talks that I've had, as far as bottlenecking and the founder effect, and losing the dominant traits from the population through some mishap, where the recessives are then able to grow in proportion to the dominant traits. A lot of that's covered in previous talks, but once you start to understand some of today's talk in relation to those talks, and I'm able to stitch it together in the coming weeks, that should all start to like coalesce in your head. Modern, I think it was modern. No, Magma. Sorry. Yeah, but I would, if you want to know more about recessives and that sort of thing, I would go back to the founder's effect, the bottleneck effect. Take some of this terminology with you and try to understand what I'm talking about with this terminology in the back of your mind, and see if that doesn't help a little bit.
Yeah.
See if somebody suggest maybe seems like everybody is interested in that recessive genetic. I mean, I guess, I don't know, like most of the good stuff are recessive, seems to be, or most of the stuff that everybody's chasing after seems to be the recessive stuff.
That's where the novel stuff is. I would agree. That's where the unique traits are going to lie in any given line.
Yeah, unique's a good word for it. Because sometimes they're good, sometimes they're terrible.
Yeah. Well, yeah. Recessive doesn't mean bad. It just means it needs a bit more of a it needs a more unique set of circumstances to show up.
Yeah, and sometimes it'll have like unforeseen possibilities where it might be seen as good in one instance, but you go down that genetic line and you find issues with it. Like everything that has genes related to that trait expresses something like it's powdery mildew, like it's prone to get powdery mildew, for example, but it happens to grow really potent medicine. So I think we can all think of some stuff where those traits are there.
Especially with the recessive traits become, well, for it to be expressing itself, it's had to become homozygous. So in the line, if it's always there, if it's become stable, it's become a homozygous trait. So the unfortunate thing is you've now got it for good. You're not getting rid of it. To have recessive genes in the pool is fine, because occasionally it's good. It's handy to have something that might not grow above or something that needs to grow 14 feet tall. That could be the recessive trait, is growing 14 feet tall. But the rest of the
Yeah.
time, 3 feet's fine.
I've seen that in a Pakistani line. They were all short, squat indicas, and the unicorn phenotype was something that grew. I mean, you could watch it grow. I didn't understand what I was looking at. I was very new to this whole thing back then. And I looked at all the plants and I said, oh, that one doesn't look like the rest. And guess which one I called? Come to find out, there was several people hunting through those packs of those seeds looking for that phenotype. And I was like, oh yeah, here in mulch,
Uh,
had I known.
So it just goes
Yeah,
to show the importance of knowing your recessive traits when looking into lineages. Yeah. I think it's super important to cover all these things because I myself looked over all that stuff starting off. I was like, oh, okay, I'll just get seeds. I don't want all I knew is I didn't want to mess with clones at the time as a beginner because my market was very unclean and I didn't know what I was getting. So from there, yeah, it's just a lot of unknowns. So it's nice to have little things like this to really clear up everything.
It was like when I was looking through when I was looking through those Arise, I looked through some Arise, and there was one that was, what Jeff called kind of the outlier, or the kind of the recessive, and it was completely different, real spindly. It wasn't the stretcher, but it got really stretchy. Yeah, I got rid of it real quick just because I knew it was going to be a mess and it wasn't what I was looking for. But I mean, it will pop up here and there, but I mean, I looked through some things, and it popped up in there, but it was just one. But there was probably, I think really I found probably 4 distinct phenotypes, and yeah, because I had males and females out of it, so I saw a lot of both sides, and I saw, I definitely saw brother and sister plants, that they're basically going to be the same pheno. If the male was a female, it was almost identical to it. So that's kind of cool seeing, all the different genetics kind of lining up on the male and female side, and seeing how things kind of relate like that. So it was really interesting. But yeah, I found a crazy outlier, and it was completely different, and it had to have been something definitely recessive, more of a sativa leaning, probably like that Hawaiian sativa that's in the Arise.
Yeah, I've seen the question from Chad about recessive traits becoming dominant and then a new recessive appearing in the gene pool. The only way that really comes to mind is through mutation, where you select for the recessive and then the recessive becomes the only option. If you were to shift the environment into a situation where that recessive only option is no longer favorable, genetics is going to try to mutate. It's going to try to pivot, essentially. Mutation is probably the quickest, easiest fix. And that's the only plausible way is coming to mind for a new recessive to appear in a recessively dominant situation. That's funny. Recessively dominant. But where the recessive is the only option, nature's pivot to that is mutation. So that's the answer there. You could do it, but you would have to try to look for, enhance, or select for that mutation to then try to find that new recessive, so that's how speciation kind of occurs, is you get a recessive or some particular genetic, and then the environment changes and that genetic's not favorable, but it can't go back to where it came from. It's got to keep going forward. So it has to change more. You do that enough times and the changed genetics can no longer mate with the genetics they originally came from, the original dominant genetics. Those two are no longer compatible. You have a new species. That's one way that happens. The other way that it could happen, not the only other way, but you could dominant to where the dominant no longer communicates sexually with the recessive, and you have speciation again. So
There's also one of those that, I mean, that's like an incredibly lengthy process.
Yeah,
Well,
we're talking.
Yeah.
I mean, you think about that's kind of how, cannabis started, really. I mean, it really started from one plant, and when it traveled, it kind of, did like you said, basically
chilies.
it just kind of adjusts its way out, and it doesn't revert back to those old genetics. It just keeps mutating forward, and it did it here, and it did it there, and it did it here. You know what I mean?
Jade was asking in chat, if he S1 a plant, would it bring out recessive traits?
Yes.
Yeah.
Yes. S1 a plant would bring out. That would create an S2. S1 being the exact same plant that produced the pollen is the one that you're pollinating.
But yeah, the donor and the receiver are the same.
Because if it has any hidden recessive traits, they are now likely to be really amplified. You've just taken a cup, so you said you have an option of, I'll bring the two recessive traits together. Because really, we've been seeing earlier. If it was really heterozygous, it's now homozygous.
Really, when you S1 something, you open it up more than what you were trying to lock it in. And S1 is pretty comparable to like an F2 generation, isn't it? I mean, you're gonna see a lot. You're gonna see a lot. I mean, if you run a big enough selection, you're gonna see a lot of the parents and stuff come out of those, are like the parents and the grandparents come out of those S1s, right?
An S1 is equivalent to an F2 in a normal crossbreeding line, yes, because that's where you've taken. Normally, it's all your F1s that you're crossing to give your F2s. Your S1 is the result of pollinating this thing with itself.
Within the same line.
Yeah. Right.
With itself. Right. S1 is within itself.
Yeah.
If you cross it with another plant, that's an R1. If in the same line. So if I took the S1, if I took the pollen from this female and pollinated another female from the same generation, that would create the R1 generation. Because that one has a different genetic makeup to this one over here. And so that just greatly amplifies. So it's a good way of finding out how stable the line is how much variation do you suddenly see in that S1. If they all come out looking pretty much the same, then you know it's a very stable line. They've worked out most of the recessive traits, or the they've worked out the heterozygous traits, and it's made predominantly homozygous traits that are sitting in there. If you get a shit ton of variation, then you know it's not a very stable line. It's kind of a one-trick pony. Hard to produce.
I'm gonna have to play with that a little bit because I think it's only the same on paper. I don't think it's the same in real life. Like in actuality. I think the math is the same between an F2 and an S1, but I don't see it giving the same results.
It's got more variety than an F1 generation would have if it was a true F. But this is the thing, is the F1 generation is just, I mean, there's variety in it unless it's two work lines that are brought together to produce a dihybrid F1. You always get this variety in the F1 generation. We're not bringing together truly distinctive familial lines each time we create an F1. We're just bringing whatever together. So the problem is these days your F1 has variety in it. But if I brought two homozygous stable lines together, I'd wind up with homozygous. Well, no, this uniform expression in the F1s. And then the
Right.
F2 would suddenly, boom, explode with the variety.
Right, like crossing like Deep Chunk and Cindy 99. You're gonna get that.
That's the plan, buddy. That's the plan.
You have like 3 phenos. Like here's D chunk, here's Cindy 99, here's a hybrid. That's what you got.
Yeah, you might have a couple different hybrids. So you might have like 5.
Couple leaners, yeah.
You have a one lean in one way, one lean in the other way, and one just kind of straight up in the middle.
Yeah.
But you pop enough seeds, anything's a gradient. You know, there's a whole schmear from one end to the other.
Good use of the term schmear.
Schmear, yeah.
Schmear.
Traditional schmaltz, butter, cream cheese, you know, your schmears.
Also, last call at the bar.
Well, I'll say this about speciation too. It's kind of interesting because some work that I've read is about when cannabis and hops differentiated. It was one protoplant, we believe, so it was one plant, and then it was through glaciation that it ended up getting separated, and then what we believe turned into hops had to develop a rhizome survival strategy, and that's really the biggest difference is the growing, you know, morphologies and some of their productions. But looking at their protoplant, I guess some Germans did a lot of really interesting research on it. Just kind of neat to look at the history of where we think that came from, because they were able to look at it from their perspective. So it didn't have any of the, you know, taboo-ness or, you know, anyone not wanting to mess with it. So it was pretty neat.
Yeah, that's pretty cool. When I was learning about speciation, we all got assigned an animal. We were in animal biology learning about this, and I got assigned a salamander. And digging into the depths of that, it was cool. The salamanders started off as one population, and a friggin' mountain range grew up right in the middle of them, split them in 2, and now they're different species. So mountain ranges, glaciers, bodies of water, those are good examples, good thought processes of how speciation occurs as you get this permanent divide between 2 species or between 2 subsets of the same species. But we're doing that these days by sending 10 packs of seeds worldwide as well. So
the Galapagos Islands is another good example of it.
Yeah.
Mhm.
Yeah, the finches of the Galapagos in particular. There's like 5, I think. They all have different beak shapes based on how they eat. It's fascinating stuff.
Yeah, some are insect eaters, some go for nuts, some go for fruits. So you'll exactly like you were saying, the shape of the beak will show you like, oh, this is an insectivore, just because it needs to crunch down on those kind of shells.
Haven't iguanas there evolved to eat a vegetarian diet too? Like they'll dive down, swim, and eat like sea plants and stuff like that. It's
Kelp,
kind of weird for an iguana. Yeah.
Goddamn hippies.
Yeah,
That Trini chicken, huh?
Being able to wrap your mind around the vocab today is really the basis of being able to talk about all this other cool shit. It's the boring necessity. It's the drudgery of it all. It's the vocab and these fundamental processes that you can't actually see. We can see chromosomes, and with the right freaking microscope, you can see DNA and such, but the layman's not ever going to see that stuff. We just need to understand the basis of it to know that what's going on with what we can see, because that's how most of us apply things, is what we can see, what we can interact with. So I look forward to getting into those types of topics, but it's a necessity to understand this deeper level molecular stuff first.
Yeah. And then one more comment that's just from Magma. He was saying Burbank was a master of this. So I'm pretty sure he's just talking on the speciation.
Luther Burbank was pretty famous. Not as famous as he should have been. breeder. He made over 800 different cultivars of fruits and vegetables in his lifetime. So fascinating dude.
Can't
That
wait
was
till things
easier back
open
then.
up.
There was fewer
I can
things
visit.
muddying the waters. Hey, I just crushed these two together. I've got a new line.
He's got an
Hey, I
incredible.
got a seed bank.
He's got an
Didn't he
incredible
come up with
blackberry.
Didn't he invent sour banana tangy or something?
Guava dog. But yeah, just like
He knows what's in
you
cookies,
always,
man.
just like you
The
always saying
original
earlier, you
cookies.
know, the
He
fucking
did it.
the lake divides the fucking population, and then they both bottleneck off, and then one human just comes in and take one from one side and one from the other side, cross those motherfuckers, and boom.
He
You
did
know,
make he made a blackberry that's white. The berry itself is white. I'm still trying to get that cultivar in my hands. It's so cool.
white but identifies as black.
Right.
Fuck them. What a berry.
Woke
No,
berry.
it's really cool though. It's pure white. Pure white rhubarb.
All right. Well, tell you what. On that note, I think we'll bring the show to the live part of the show to an end. We'll have a little after show party, which I'll share the link in a minute. But yeah, GMF, where everybody can find you, sir.
You can just find me hanging out here at Operation Grow. You can find me hanging out with the neighborhood. We're usually on Mondays at 4 p.m. Pacific, 7 p.m. Eastern. You can find me really just, anywhere in these chats, hanging out with all the wonderful people that, I've been lucky enough to meet. Like, I know this sounds weird, but every day I'm grateful for the fact that I've gotten to meet all you people. And this is kind of like the first year of really, for lack of a better term, started living online, getting to know people I didn't actually know in real life. So I'm very, how should I say, grateful for the community and grateful for people like you, Pete, that make this platform. Grateful for people like Cascadian that, share education for everybody. So, I just, if I can be helpful, I do a lot of food science and ferments. Hit me at Genetic Memory Farms on IG. But, thanks, Pete. I really appreciate you making this possible.
Yeah, absolute pleasure. And Duckweed, where can we find you, buddy?
Hey, you can find me here, Operation Grow, of course, and on the Neighborhood Grows, which is usually on Monday nights for me, 7 p.m. Eastern, 4 p.m. Pacific, I think. And on Instagram, NutsandGuts868. Yeah, cheers everyone. Thanks for coming out, and thanks Cascadian again for this wonderful presentation. Bless up.
And Lefty, buddy.
You can find me at Lefty's Farm on Instagram and Cannabis. And thanks again to Cascadian for that great presentation. Like I said, it was a good refresher for me, man.
Yeah, it certainly took me back, that must say. It was good to have that yeah, jogged some old memories. And Raptor, where can we find you, sir?
Hey everyone, great show. Learned a lot. Great refresher for myself, and just reminds me to bookmark all the books that I've saved throughout the years of learning this stuff. So yeah, I'm Raptor Grow. You can find me at Raptor Grow on Instagram. The Neighborhood Grow Show on YouTube. Check out our Instagram as well. Just like Duckweed was saying before, 4 p.m. Pacific Standard Time, 7 Eastern, and 1 o'clock UK, or 1 a.m. UK. Be very specific. We're working on it, Pete. We'll set up a nice recording episode for you so you can be included. No worries. But yeah, you can also find me here at Operation Grow. Same time, actually, no. What am I thinking? 1 p.m. Pacific Standard Time, 4 p.m. Eastern, and 9 o'clock UK time, 9 p.m.
Yeah.
So yeah, hope to see you all soon.
Cool. And Cascadian, thanks again for the presentation. Really well. Went that well. Very much appreciated. Where can everybody find you, buddy?
You can find me if you have questions. Definitely hit me up today at Cascadian Grown on Instagram. I'll just be chilling, trying to field some questions. Hopefully you guys come find me. You'll find me here, bending your brain and mine, apparently. And I think that's all I'll commit to right now.
Cool. And,
Cascadian Hort.
you'll be able to find myself at Operation Grow 2019 on Instagram, PL Grow Systems as well on Instagram, as well as plgrow.systems for Reminder Unit. And yeah, on that note, hang on, what am I trying to figure out how to do here? I'm trying to play out with the intro as well, but I don't know how to mute everybody, so we're just gonna have to keep quiet. Nobody say a word. Yeah. So thank you once again, everybody, and we will catch you all next week on the Operation Grow Show. Stay safe and keep well.
Peace out.
We'll be right back after this important word.
A joint. Ever had one? It really sets you up, especially at breakfast time. That lovely mild flavor. Gets you up quicker than anything that does. Up and away. A good joint sends you off to a flying start. Puts you way out ahead of the rest of the crowd all day. Gives you energy to keep you going. Joints.